This is podcast number 47. We're going to continue our discussion of emergent properties with another example of an emergent property, and that's a feedback loop. And you've seen this before when we talked about the Lac operon, how the product of a pathway can go backwards and affect the pathway, turning it on or turning it off. In this one, there's a little more complicated, and it talks about a virus. This is the lambda-phage, the bacteriophage. This is an electron micrograph of the virus, and this is just a cartoon. This is the head where the nucleic acid is. Excuse me. That's the tail through which the nucleic acid is injected. The arms are where it attaches to the bacteria that it's going to infect. Okay. Here is a heavily infected cell. Here you see all these little heads and then some on the surface of the cell here. Normally you wouldn't see so many, but this is in a phase of the virus reproduction cycle where it's about to burst. You can grow viruses, phage viruses in the lab on agar plates. This would be E. coli grown as a lawn so that the cells are so dense on this lawn that they are so dense that they're touching each other and it looks a little fuzzy. That's why it's called a lawn. And then the virus forms what are called a plaque. And this is virus has reproduced inside E. coli cells and then burst and left a clear area on the plate. So there's high concentration of virus there, and it's clear because it's killed the bacteria in that area, and that's called a plaque. So when these bacteriophage enter the E. coli, there's one or two ways to go. They can inject the genome, and then the genome here in red can become part of the bacterial genome, the circular chromosome here. And then every time the bacteria replicates, it also replicates the virus. And so the virus can reproduce in that manner. It's relatively slow, but it's still exponential growth. You know that bacteria can grow quickly into large numbers. If you've ever had a can or a jar of spilled milk or spoiled milk in your fridge, you know it can be fine one day and then totally full of bacteria the next day. And if it goes in this direction where the virus incorporates itself into the genome of the bacterium, that's called lysogenic, like Lysol, lysogenic. But it can also go in a different direction where it will use the ribosomes of the host cell, the cell that is infecting, to make new copies of the virus, and then assemble those new copies, and then the virus will burst that cell that it infects and kills the original cell. So, and that's called lytic for lysis or split, Lytic. So, what causes this virus to decide, quote unquote, to be in the lysogenic phase or the lytic phase? That'll be the focus of this podcast. It's not really making a decision, but it can go either way, and the information it gathers and the rules by which it goes one way or the other way are some of the emergent properties that we're going to talk about. So this is discovered in the 50s that if you treat a cell that is in the lysogenic phase and that has incorporated viral genome in it, it can get activated by UV light, and then that will cause the virus to exit the lysogenic life cycle and go into the lytic life cycle here, where it replicates and then bursts the cell. If you look at bacteria infected with cells in a population of bacteria, there's always going to be some that are on the lytic pathway and some that are on the lysogenic pathway, because these pathways have a little background leak and a little bit of randomness to them, so that they're the right molecules bumping into the right places on the DNA randomly can cause things to happen, and that's why it's not exactly 50-50. But if there is a loss of lysogenic cells, the bacterial cells will form colonies. So if there are few cells that are infected in the lysogenic phase, the cells, bacterial cells, will form colonies. If there's a lot of lytic cells, you'll have more plaques formed, and you can see both sorts of things happening on a single agar plate, depending on the conditions. So, what are the molecular rules that cause the virus to go in one direction or another direction? So, hopefully this looks a little familiar. We had a similar sort of diagram when we were talking about the lac operon. And lac operon was an example of how a cell can get input from its environment and decide, quote unquote, what to transcribe into mRNA and then translate into protein. In that case, we saw operators and promoters and that sort of thing. These RNA polymerases bind to promoters, okay? Either promoter RM or promoter R. They are facing back to back to each other. Promoter RM translates in this direction, off to the left as you look at it, and promoter R translates off to the right as you look at it, and they translate different products. The one promoter R produces an mRNA and then a protein called CI, and then the one off to the right here produces an mRNA and then a protein called CRO. There are three operators, and you would not ever see two RNA polymerases bound to these at the same time, and the reason is that these operators are not evenly distributed. So operator 2, the one in the middle, is positioned so that more of it is to the right and less of it is to the left. And so either the RNA polymerase, which covers all of operator 3 and a little bit of 2, or operator 1, which covers all of 1 and a little bit of 2, they both have to cover a little bit of 2, and so you can't have both of them at the same time, and hopefully it'll become obvious as to why. So if this promoter is active, you make that message in that protein. If this promoter is active, you make this message in that protein. And if you make this protein or this protein, determines whether or not the virus is going to go into the lytic phase or the lysogenic phase. Okay. The protein that is made on this side, on the CI side, is called the CI protein. And it comes as a monomer that has two different domains, the C terminus and the N terminus. N terminus is where the protein starts, C terminus is where it ends. Two monomers together can make a dimer, where they bind together. Two copies of the protein binding together. And there can be an equilibrium between these two states. It can go from here to here or from here to here. The dimer binds to the operator on the DNA and acts as a transcription factor. So transcription factor, in this case a trans-acting transcription factor, binds to the DNA and then changes what's made. So it binds to the DNA in a cooperative manner, in that it has a preference as to which binding sites on the DNA it'll bind first, and then second, and then third. In the case of the CI dimer, the product of this promoter, it binds to the first operator first, conveniently, and then to the second operator, and it says quickly. So this is cooperative binding. Binding to the first operator, which is the highest affinity, or the DNA likes this, or the protein CI likes this spot on the DNA the best. Think of it as the strongest attraction, like a magnet. Okay, so 1 always goes first, and then once 1 is occupied, 2 is quickly occupied because of cooperativity, similar to what we saw with hemoglobin. Once you bind 1 oxygen, it makes the second one and third one, fourth one easier to bind. Okay, so you bind 1, and 2 binds quickly. The emergent property here is cooperativity. We saw that again when we saw how hemoglobin works. Okay, and then once 1 and 2 have CI protein bound to them, now RNA polymerase can bind to the PRM promoter and chug away and transcribe CI mRNA, and then translate that CI mRNA into protein. Okay. If you have different amounts of CI made, it causes different effects. So a little bit of CI, operator 1 gets bound. A little bit more CI, operator 1 and 2 gets bound, and then the promoter here can be occupied by RNA polymerase, and you make more CI. If you make too much CI, though, operator 3 also gets bound. Operator 3 has the lowest affinity, so this is the weakest magnet. It always gets bound last, and once it binds, it shuts it off. So too much CI blocks the CI production. So if operator 3 is bound, now RNA polymerase cannot bind, and you don't get any more CI being made. Okay, so there's a feedback loop in that the product of turning on this operator makes a protein, which will come back and turn on the operator even more. Eventually, it'll make so much protein that it turns off operator 3, and RNA polymerase can't get on operator 3, and it shuts off production of this transcription factor. Okay, and that's going in this direction. Okay, and this would cause if this promoter is active, this would cause the virus to be in the lysogenic phase. And lysogenic phase, again, is when it incorporates itself into the genome and then hangs out on the bacterial genome and just replicated when the bacteria replicates. Operator 2 and operator 1 are occupied. Also means that the other promoter, PR, that makes this other gene product, CRO, cannot be occupied. So when these are on, that's required for this one to go off to the left, but it also shuts off the one to the right. And that's why you can't have them both occupied at the same time. Okay. And too much CI also blocks CI production. If you turn this on for so long that you get a bunch that it finally binds to the weakest magnet, operator 3, then that shuts off this side of the promoter. Okay, so how do you turn on the other side of the promoter? Well, way back here, I told you, but I didn't really show you on the graph, that UV light can cause the cells to go into the lytic phase. Okay, you turn on UV light, and cells will go into the lytic phase. Okay, so how does that happen? What UV light does is it activates a protease. The protease is made by the bacterium, and that protease affects the transcription factors that bind to those promoters that we were just talking about. So why would the cell make protease? It could be activated by UV light. It could also be activated by, there's lots of protein around for the bacteria to eat, and that is the same as lots of bacteria for the virus to invade from the virus's point of view. So that would be another signal to become into the lytic phase. I'm a virus, I'm hanging out in the lysogenic phase, I'm replicating but not at a really high rate. Now I find out that there's lots of protein around. If there's lots of protein around, there's probably a lot of bacteria around to eat it, a lot of targets for this virus, and so I'll go into the lysogenic phase, go into the lytic phase, leave the lysogenic phase, go into the lytic phase, release a bunch of viruses, and reproduce like crazy. Okay? So the protease that's activated by UV light can cut up this CI protein to a point where it can't bind to the operator. Also will break dimers into monomers. Okay? And then that means that operator 1 won't get bound, operator 2 won't get bound, and the PRM, the left side of the pathway, won't get activated. So if these are empty, now the other side, CRO mRNA, can be transcribed because now RNA polymerase can bind to this side. Okay, when they're occupied by these CI dimers, RNA polymerase binds to the left side and goes off, and here makes CRI. But if they're not occupied because the protease cut them up, now operator 1 can go off to the right and make CRO RNA. The protein made by CRO mRNA is CRO protein, and it comes as a monomer and also as a dimer. That dimer will bind to operator 3 first. Okay, so when this protein forms a dimer, it binds to operator 3. Remember, that was the third choice for the CI protein, but for the CRO protein, it's the first choice because the CRO protein needs RNA polymerase to bind to 1 and 2. So it needs 1 and 2 to be empty. I'm going to fill up 3 with CRO protein, and I will go off to the right and transcribe my CRO mRNA, turn that into CRO protein. Okay? And just like with CI protein, it depends on how much you make. So at first, you make a little bit of CRO protein, binds to operator 3, and stimulates the production of more CRO mRNA. Now more CRO protein gets made. Operator 2 gets some bound to it, and then even more CRO protein could bind to operator 1. Both of that will block more CRO production. Okay, so this also has a feedback loop in that if you make too much, it'll shut it off, but just a little bit, and it'll stimulate it. Okay, another feedback loop. Okay. So this lambda switch, if the virus is in a lysogenic lifestyle, this is misspelled, sorry, SDYLE. If it's in a lysogenic lifestyle, then the virus is incorporated into the bacterial genome, and the virus only replicates when the cell replicates. In that case, there's a high amount of CI protein and a low amount of CRO protein. High amount of production of the promoter going to the left, low amount of the production of the promoter going to the right. In the lytic lifestyle, the opposite is true. There's a low amount of CI protein because you've destroyed it because you activated the protease, okay? Or there's just a lot of protease around because there's a lot of bacteria around. And then there's a high amount of CRO protein. Okay, so the lambda phage, this virus is not thinking. Okay, it's not thinking about, oh, the conditions are right to leave my bacterium. I should replicate and destroy this ship, and then I'll chew up a lot more bacteria. It's not thinking at all. But it can have this emergent property in that it's making quote-unquote decisions about its environment. Is this a good environment to replicate, or is it a good environment to hang out and see if conditions get better? If UV light is damaging the host cell, that's not a great situation for the virus either, because that UV light is probably damaging the host genome, and the virus is part of that host genome. It doesn't want to get damaged, so it's good to leave in that case. So UV light can stimulate a pathway that'll cause the virus to replicate and then leave and find a new cell. If the host cell is well fed, so it's well fed, there's a lot of protein around for the E. coli to eat. That's a good signal for the bacterium to make more copies of the virus because there's more targets out there, and then the virus can grow and replicate. And so that's another good signal, reason to leave lysogenic and enter lytic. So again, it's not that this simple virus is making decisions, but decisions are an emergent property of these molecules coming together and operating in the way that they operate through positive feedback loops.